Hello everyone, this is Dr. Pooja, your biochemistry mentor. Now in this present video, we are going to discuss a very interesting and a very important topic in biochemistry. Now we are going to discuss about glycogen metabolism. A very important topic. In fact, all the topics are important, but this is again a very important topic. Now see, first of all, you should understand what is this glycogen. Okay, glycogen is the reserve food or the amount of glucose in the liver as well as the muscle. As I've been, in my previous videos, I've been discussing about the well-fed state, about the fasting state, about starvation. So how our body meet the energy requirements, and keep one thing in mind that our heart, brain are the two organs which just cannot stay without energy for even a parsec of second. And brain though it always relies on glucose for energy during the fed state and the fasting stage. Okay, if we talk about RBCs again, since they have to again circulate blood from head to toe, they also have to rely on glucose for energy. Now the question is, what is this glycogen? Why body has to store glycogen? How this glycogen is metabolized. So this glycogen metabolism we can actually summarize or we can study under two subheadings. One is the synthesis of glycogen, which is known as glycogenesis. Another process, because obviously for one process there has to be an antagonistic process. This is the actually beauty of the body. So glycogenesis ka reverse antagonistic process is glycogenolysis. Again, you should know the basic concept of lysis and genesis, which I have been discussing so many times in my other videos. So if we talk about genesis, we can simply say that जेनेसिस है तो इट इज सिंथेसिस. ग्लाइको तो सिंथेसिस ऑफ ग्लाइकोजन. अब फ्रॉम वेयर ग्लाइकोजन इज सिंथेसाइज्ड, सो द प्रिकर्सर इज ग्लूकोज. So I can simply say that glycogenesis is a process of synthesis of glycogen from glucose. अब since it is synthesis, तो is anabolic process. See, all the things are interrelated with one another. Now coming over to glycogenolysis, which means breakdown of glycogen. Now coming over to this, since the precursor of glycogenesis is glucose, so glycogenolysis will always be the degradation of glycogen to glucose. So I can simply say that glycogen will be released to form glucose. Now genesis is an anabolic process, so glycogenolysis is a catabolic process. And then fourth very important point is, now after seeing all the videos on carbs metabolism, now you people should be able to differentiate between genesis and lysis, that glycogenesis is occurring in which stage of the body and glycogenolysis is occurring in which stage of the body. So if I talk about genesis, you can very well write it on your own that it occurs in the well-fed state. And if we talk about glycogenolysis, it occurs during fasting. Or if we talk about the fasting, it will start after the post-absorptive stage. So it will be early fasting, you can say till 16 hours, the glycogenolysis will be the one holy solely source of glucose to the body. Then coming over to this, question is, have you ever given a thought, because we've been always studying for so long, for so many years, that fats are the instantaneous source of energy. Okay, so what is the difference between actually the beta oxidation of fats and glycogenolysis? Why? What is the use of storage of glycogen in the body. So the question is, why body stores as glycogen? Has to store this. Right. Now the most relevant answer to this is glycogen can be broken down anaerobically. We never say it is oxidation of glycogen. We always say it is the breakdown of glycogen. Glycogenolysis is the breakdown of glycogen. Okay. So we will always use breakdown. We will never say that oxidation of glycogen is there. Getting my point? And if we talk about the fatty acids, we always say beta oxidation of fatty acids. So the most important difference lies here. Glycogen can be whenever anaerobic condition is there or in the absence of oxygen, and that is what is happening in the muscle also. Okay, that is another thing key in the muscle. Glycogenolysis will never contribute to glucose directly because of the absence of the glucose 6-phosphatase enzyme. But yes, the most important one is glycogen can be easily broken down without requiring oxygen. This is first very important thing. So I can simply say that glycogen does not require oxygen for the breakdown. Whereas fats will always require oxygen for their breakdown. Right. Then, second important point is that glycogen, we have studied the structure of glycogen in detail in the video on polysaccharides. So we can say that glycogen has reducing end as well as a non-reducing end. So if we talk about glucose and glycogen, glucose is readily soluble in water, whereas glycogen is very least soluble in water due to non-reducing ends also and reducing ends also. So glycogen, even if it is stored, it is not going to disturb the osmolality in the body. So glycogen cannot disturb the osmolality of the body whereas glucose can. Because glucose is highly soluble in water. So if I say that glucose is highly soluble in water, so obviously it is going to disturb the osmolality. And we can say that since it is soluble it will disturb the osmolality, thereby leading to swelling and death. Right? So we can say there are two main reasons. One is that glycogen can be easily broken down anaerobically, whereas fats cannot be broken down anaerobically. Second is glycogen cannot disturb the osmolality, whereas glucose can. And the reason behind this is because of the structure of glycogen. So the body always stores glycogen. Now the question is where glycogen is stored. So always remember the storage of glycogen is stored in the liver and the muscle. So there are two organs for the storage of glycogen. And if we talk about the total composition, how much is stored in the liver, how much is stored in the muscle. So always remember, if we talk about percentage by tissue weight, it is more in the liver. But maximum storage is in the muscle. So it can store up to 10% in the liver, whereas 1 to 2% in the muscle as per the tissue weight. Okay, so I can simply say that glycogen is occurring in the liver as well as the muscle. Now, the question is, first of all, what is glycogenesis? How it occurs? What are the different steps of glycogenesis? And what are the steps of glycogenolysis? So first of all, we are going to take these processes one by one. First, we are going to take up glycogenesis. Glycogenesis. Now see, important points about this is glycogenesis, synthesis of glycogen. I'll just quickly summarize this. Synthesis of glycogen from glucose. Second important point is cytoplasm. Because site, I told you, it plays a very important role. Site is the cytoplasm. Where? In the liver as well as the muscle. Third important point is, since it is an anabolic process, right, increased in when the body has high insulin to glucagon ratio. Because obviously when the ratio of insulin to glucagon is high, that means increased amount of insulin is there, and insulin always promotes the uptake of glucose. So excess amount of glucose will be stored to form glycogen. This is very important. Or you can say that since it is occurring in the well-fed state. Now, the question is that what is the regulation of glycogenesis, or how glycogenesis is regulated. So always remember, if we talk about regulation, the rate limiting enzyme of glycogenesis is glycogen synthase. It is not synthetase, it is synthase. Glycogen synthase is the rate limiting enzyme. Okay, and I just want to mention it, synthase, not synthetase, because I have told you clearly the difference between synthetase and synthase, that synthase mein no ATP is required, whereas synthetase cannot proceed further without ATP. So here simple synthesis of glycogen will be there, and it is a very, very simple process. Now the question, this is regulation. Now the question is, what are the different steps of glycogenesis? Now if we talk about the steps, the first step is synthesis of UDP glucose. The first step is synthesis of UDP glucose, or we can call it as UDPG also. Okay, or this is C1 activated glucose. C1 activated glucose. Now what happens is, again, if you go back, this is actually, we are just activating carbon 1, because we know that glycogen is nothing but simply alpha-1,4, alpha-1,6 glycosidic linkage. And since it is a glucosan, so it has all the identical glucose molecules which are linked by alpha-1,4, which are linked by alpha-1,6 linkage. Now the question is, how this UDPG is synthesized? सबसे पहला initial step would be simple, as we've been studying. This glucose will undergo phosphorylation to form glucose 6-phosphate. The name of the enzyme here is hexokinase. We all are familiar with this enzyme. We have discussed this enzyme in detail, or we can say ATP to ATP. Now, this glucose 6-phosphate by a simple interchange reaction, it will be converted into glucose 1-phosphate. Means from 6th position, it will jump to 1st position because we have to activate C1 of glucose. Why? Because we have to make a glycosidic bond. And here the enzyme is phosphoglucomutase. And it is going to form glucose 1-phosphate. Then activated, so UTP will come. And it is going to form UDPG, which is uridine diphosphate glucose. And here the name of the enzyme is simple, you can say UTP phosphorylase, or if we have to write the expanded form, uridine diphosphate pyrophosphorylase. Whatever you can write, uridine phosphate pyrophosphorylase. So we can simply say that this glucose will be converted into UDPG. So this becomes the first substrate of glycogenesis. Okay. Then the second step is, now see, we know the structure of glycogen is what? It is having alpha 1,4, alpha 1,6 linkage, and just keep one thing in mind, it is not possible to put everything in one go. So it has to be stepwise, and the body just cannot spend so much of time just to phosphorylating one molecule again and again. So there has to be a primer which is already synthesized in the body, which can just help the body in the reactions. So the second process is the formation of a primer. It's a very important step, formation of a primer. Okay, and this primer is actually known as glycogenin, very important. So glycogenin, which I can say it's simply say 40 to 50 glucose residues long, depending upon how much glucose has to be converted into glycogen. It can be 10 to 20 also, it can be 20 to 30 also, but it is just acting as a primer. So I am just giving you for example that 40 to 50 glucose residues. Right, synthesized on the backbone of the polypeptide. Synthesized on the polypeptide backbone. And here the activated residues are tyrosine residues. So glycogenin will come and it will simply help in alpha 1,4 glycosidic linkage. So I can simply say that, say if this is glycogenin, right, it will simply help in the formation of glycogen like this. Now the thing is that we have to make alpha 1,4 linkage. So there will be two activities at the same time. Glycogenin is the primer, and along with glycogenin, glycogen synthase will come. So this glycogen synthase is going to help in the synthesis of alpha 1,4 glycosidic linkage. Right. So with the cumulative effect of glycogen synthase, we can say the entire molecule will be synthesized like this. Now the question is, this is alpha 1,4 linkage. Glycogen is having both 1,4 as well as 1,6 linkage. So the third step, which is actually the final step, would be the role of the branching enzyme. So this branching enzyme, as the name suggests, it is going to incorporate branches, and it is going to incorporate alpha-1,6 glycosidic linkage. And the structure would be like this, again like this, 1, 2, 3, 4, 5, 6, 7, 8, and it will be like this. And this is how the entire molecule of glycogen will be synthesized. And we can say that this is alpha-1,6 and this is alpha-1,4. Right? So, with the help of glycogen synthase, with the help of branching enzyme, and with the help of glycogenin, the primer, the entire molecule of glycogen is synthesized. And always keep one thing in mind, that DNA replication and glycogenesis are the only two processes in our body which requires primer. DNA replication and glycogenesis, only two processes requiring the primer. Here it is glycogenin and DNA mein again the RNA primer is there, which is synthesized as a complex known as primosomes. Later on it is removed. Okay. So this is a very important point. DNA replication and glycogenesis. So this is what is glycogenesis occurring in the well-fed state. It's an anabolic process, or the most important point here is that it is always accelerated in response to high insulin to glucagon ratio. And obviously you can say that insulin is going to accelerate because it is an anabolic hormone. Okay. So regulation we will be discussing along with glycogenolysis. So here I can simply summarize it that the rate limiting enzyme is glycogen synthase. Right? And glycogen synthase is occurring in two forms. This we will be discussing again in detail, active and inactive forms. So, abhi for your knowledge sake, I will just write the diagram, draw the diagram of active and inactive. So, active is like this and inactive is like this. So, I can say active is dephosphorylated because it does not have phosphate, and inactive is phosphorylated because it has phosphate. Active, inactive. And obviously you know what is the meaning of active. Active hai to glycogen synthesis hoga, inactive hai to glycogen ka synthesis will not take place. This is all about glycogenesis. Then the parallel process here is, the second one is glycogenolysis. Now see, when there is a dearth of breakdown of glycogen, when there is a requirement of glycogen, obviously when the body requires energy, तो सबसे पहले glycogenolysis will be simple breakdown of glycogen. अब breakdown of glycogen will be what? It will be broken into glucose, obviously from where it is synthesized. So breakdown of glycogen to release glucose. Okay. Now see, here I will just write one thing. Here you can say that the entire glycogen is synthesized. Now it has to be broken down into glucose. And when it is broken down, or to what it is broken down? When it has to release glucose. Okay. So Glycogenolysis will be like this. Since it is breakdown, so it has to be a catabolic process. Most important thing. And it will be favored by low insulin to glucagon ratio. See, everything is in parallel. Glycogenesis is like this, so glycogenolysis is like this. If glycogenesis is occurring, there is no question of glycogenolysis and vice versa. So, if glycogenolysis will occur, there will be no question of glycogenesis. So, decreased insulin to glucagon ratio is there. Very important, right? Now see, we have already a molecule of glycogen. And if we talk about this, one more thing I just wanted to write the site first of all, because if we talk about the site, where the synthesis is taking place, the site means the breakdown will also take place in both. So, site is again cytoplasm of the liver and the muscle. But the most important difference between glycogenolysis occurring in the liver and the muscle is, in the muscle, glucose 6-phosphatase enzyme is absent. So, I can say that it is occurring both in the liver as well as the muscle. In the muscle, glucose 6-phosphatase enzyme is absent. Phosphatase is absent. This is a very important point to remember, right? Now, coming over to this, how this process starts, or if I have to write it into steps, what are the different steps of this? Now, if we talk about the steps, we have the first step is phosphorylatic splitting of glycogen. This is a very, very important step. Phosphorylatic splitting. Now, what do you mean by the term phosphorylatic splitting? Now see, we have glycogen with us. Glycogen is having this type of structure. Right, that is what we have drawn earlier also. This alpha 1,4 highly branched. So, this is alpha 1,4 and this is alpha 1,6. We can elaborate it further also. I am not just elaborating, I am just giving you an idea ki how it is occurring. Now see, what will happen? First enzyme will come here, or I can simply say that glycogen is having n number of glucose molecules. It can have 100, it can have 200, it can have 300 depending upon, again you can say it again varies from person to person. Now this glycogen will undergo phosphorylatic splitting, which means that one molecule of glucose 1-phosphate will be liberated, and the enzyme here is glycogen phosphorylase. Very, very important to understand. Glycogenolysis is just a game of two enzymes, that's it. जैसे glycogenesis में we required 3 to 4 steps, but here only 2 steps are there. So, glycogen phosphorylase simply will release glucose 1-phosphate and resultant would be glycogen N-1, one molecule, because this is release of glucose with phosphate. Very, very important. And that is what is the importance of this enzyme also. Glycogen phosphorylase, it always catalyzes the phosphorylatic splitting. What do you mean by phosphorylatic splitting? That it always releases glucose 1-phosphate as a byproduct, and it obviously comes under transferases group of class, but it is different between phosphatases and kinases. Kinase mein it always attaches phosphate, but this is phosphorylatic splitting. If I talk about kinase, it will be glucose 6-phosphate, but here it is phosphorylase. Now once this glycogen releases one molecule of glucose, the reaction will keep on going. Now the second step, I told you it occurs only in 2 steps. Now the second step is simply the debranching enzyme, the first and the second one. So the second may debranching enzyme will come. D. Dekho, there it was branching enzyme, here it is debranching because we have to actually break this glycogen into glucose. So we have to remove the branches. Now how we have to remove the branches? This debranching enzyme is a bifunctional enzyme. It has two activities. Bifunctional. And it has two activities. One is alpha 1,4 glucosidase and alpha 1,6 sidase along with transferase. So basically it has to actually shift this alpha 1,4 to the main branch and it has to split this alpha 1,6 linkage. So what happens is glycogen phosphorylase will keep on acting unless and until we get a limit dextrin molecule. And if I talk about this, I can simply say that, say this is glycogenin. 1, 2, 3, 4, 5 and 6. And here if we say alpha 1,6 is there, so it is 1, 2, 3 and 4. This type of structure will exist 1, 2, 3, 4. And this is glycogenin. Now what will happen is there will be transfer. This type of structure is actually known as limit dextrin. Right. Now this portion 2, 3, 4 will be transferred here. I'll just write it with the help of, I'll just color it like this, 2, 3, 4. And what kind of structure we are going to get? We are going to get this type of structure. Glycogenin 1, 2, 3, 4, 5 and after this comes this 1, other 2, 3, 4. Means just to avoid the confusion I will write it like this 2, 3 and so it will be more clearer to you 2, 3, 4 and here this 1 will be there attached. This one will be attached somewhere here 1, 2, second pair like this. So this is simply you can say alpha 1,4 transferase. This is the transfer, you can see that. And what we get is we get a linear molecule now. But now the main loci is still this alpha 1,6 linkage. This is alpha 1,6 linkage. And this is broken down by this debranching. You can see removal of the branches. This branch is removed. And if we talk about the reaction here, the reaction here will be 1 to this glucose will be liberated. So glucose plus this type of structure will be there. 1, 2, 3, 4, 5, 6 and rest of the ones will be 2, 3 and 4. This is the first one which will be liberated as glucose and this becomes 2, 3, 4. And now again the status would be what? Again glycogen phosphorylase will act. Now it is a linear molecule as you can see that. So what will be released in the end? What we are going to get? We are going to get glucose 1-phosphate. It's clear. Okay. Now this glucose 1-phosphate will now be converted into glucose 6-phosphate. Simple 1 to 6, so the enzyme is phosphoglucomutase. Up I can say that glucose 6-phosphate is synthesized. Now what will happen? This glucose 6-phosphate will have altogether a different fate in the liver as well as the muscle. So if I talk about the liver, what happens in the liver and what happens in the muscle? glucose 6-phosphate has to give its phosphate now. So the name of the enzyme here simply is glucose 6-phosphatase. Now the question is that this glucose 6-phosphatase in the liver is going to form glucose, as simple as that. But in the muscle, this glucose 6-phosphatase is absent, so no glucose, right? So I can say that muscle mein there is no synthesis of glucose, so there will be accumulation of glucose 6-phosphate in the muscle, and this glucose 6-phosphate in the muscle will lead to the formation of lactate, and that is what exactly happens. Anaerobic glycolysis in the muscle will always lead to production of lactate. Very, very important. Okay. So this is what is glycogenolysis. So first of all, let me just write, if we have to just give the gist of this, I can simply say that the rate limiting enzyme here becomes, first of all, rate limiting enzyme of glycogenolysis is glycogen phosphorylase, because there are only two enzymes here. Glycogen phosphorylase is there and debranching enzyme is there. Debranching enzyme is a bifunctional enzyme which is performing two activities at the same time. One is it is simply transferring, and second is it is removing alpha-1,6 linkage. So glycogen phosphorylase. Now as glycogen synthase, glycogen phosphorylase also exists in two forms, same, just like that. So one will be active, the other will be inactive. Now what is the difference between active and inactive? And if you draw the structures will be, now if you remember the structure of glycogen synthase, you can actually understand the regulation here. If we see the structure of glycogen synthase, the active form was dephosphorylated. Here it is phosphorylated. And inactive form, there was phosphorylated, so here it will be dephosphorylated. So I can simply write it like this, that this is phosphorylated and this is dephosphorylated. One has to understand the difference of this. Clear? So this is glycogenolysis. So if I have to just write the main points, the most important point about glycogenolysis is that debranching enzyme is bifunctional. Branching enzyme is bifunctional as it has two activities. One is alpha-1,4 glucan transferase, because it is transferring, and second activity is alpha-1,6 gluco or glycosidase. Second is phosphoglucomutase is the common enzyme of both the processes. So I can say that phosphoglucomutase is common in both the processes. The processes, which is simply glycogenesis as well as glycogenolysis. And if we'll talk about this step, simply glucose 1-phosphate is converted to glucose 6-phosphate. And the name of the enzyme here is phosphoglucomutase. So these are the two very, very important points to remember about glycogenolysis. So I can simply say that if glycogenesis is occurring, glycogenolysis cannot occur. Obviously, because when the body is under well-fed state, so we are going to synthesize something and we are going to literally store that. So whenever the body is under well-fed state, glucose will be stored to form glycogen. On the other hand, when the body undergoes fasting where the body requires energy, so this glycogen will be broken down to glucose. Both the processes are occurring in the liver and the muscle, but in the muscle, glycogenolysis will never ever contribute to glucose. Why? Because the muscle lacks glucose 6-phosphatase, whereas in the liver, aram se glucose will be synthesized. So after these two processes, then we come over to the regulation. Now, regulation of these two processes is also very, very important, because we can talk on the level of, first is covalent modification. And second is hormonal regulation. Right. And we can say role of calcium also. Calcium too. Now one more thing I just want to write here, the third point, that PLP or vitamin B6 is a component of glycogen phosphorylase. B6 or PLP is coenzyme for glycogen phosphorylase. And some 2 to 5% of glycogenolysis takes place in the lysosomes also, because after this we are going to discuss about glycogen storage disorders. Commonly we call them as GSTs. There again that concept will come. So coenzyme for this, so I can simply write this reaction here where I have written glycogen phosphorylase, vitamin B6 is, or PLP is a coenzyme. And since glycogen phosphorylase is the only rate limiting enzyme, B6 also plays a very important role in this. Clear? So now when we discuss about the regulation, first aspect is covalent modification, second is hormonal regulation, and third is role of calcium. So we are going to take these aspects one by one. So first is this covalent modification. We can call this covalent modification as allosteric modulation also. Covalent modification or modulation, whatever you can say. Modification. Now see, we have to again understand what is a well-fed state, what is a fasting state. Now how the body comes to know that the body requires glucose? Because whenever we say we have to give a morning sample, say whenever a person is diabetic, every glucose should be monitored at every interval. So we always say fasting sample should be there, postprandial sample should be there. So what is the correct estimation? It's not like that we can anytime go for fasting sample. It should be always 10 to 12 hours of fasting. It should be always 10 to 14 hours of fasting. The reason behind that is because whenever we are sleeping overnight, whatever amount of glycogen is stored, it will be actually depleted, because glycogen will be broken down to glucose and we will get energy. So morning when we get up, the correct estimation of glucose is there. Okay, so this is very important. Now the question is how the entire process is taking place. Whenever the body goes, whenever the concentration of glucose decreases, the body goes into hypoglycemia. Whenever the concentration of glucose increases, the body undergoes hyperglycemia. Okay. And if the body has to take some remedial measures, obviously when the concentration of glucose is low, we have to trigger those processes which can lead to increase in the concentration of glucose. It's but obvious, because we'll have that faintish kind of feeling. And similarly, if we have more amount of glucose, that is hyperglycemia, which will again lead to diabetes mellitus. So we have to bring the level of glucose down, so that the net fasting level of glucose should be 80 to 110 milligram per dl. Right. And if we talk about PP, it should be 130, not more than that. Because WHO keep on revising the values, but it should be 130 or 140. Right. So basically, when glycogenesis is occurring in the body, glycogenolysis will not occur. There is no question of breakdown when something is being synthesized. Vice versa, if something is being broken down, there is no question of synthesis. So how this modification takes place from synthesis to breakdown? So this is always at the level of the enzymes. Because ऐसा तो नहीं है कि it will give command to something else. Because the substrates and the products are actually liable. They are the liability to the enzymes. They have to just inform the enzyme that this has to be synthesized, this has to be stopped. So everything is done by this covalent modification. अब question is how the body comes to know. So there are hormones in the body, there are second messengers in the body also. So if I talk about hormones, insulin and glucagon are there. And if I talk about the second messengers, the most important second messenger, ऐसे तो there are cyclic AMP, cyclic GMP is there, but the most important one, phosphatidylinositol is there. But cyclic AMP plays a very, very important role in the metabolism of carbohydrates, which works under GPCR. So G-protein coupled receptor is going to give instructions to cyclic AMP that this has to be released. So ideally, covalent modification and hormonal regulation are like interdependent on one another. Very important. Okay. So if I talk about covalent modification, I can simply say that I'll just try to summarize both. Hormonal regulation mein I can write insulin and glucagon. So both are actually interdependent. Now coming over to this, what happens is there is one equation which is occurring in the body. ATP is forming cyclic AMP, which is a very, very important second messenger, and this cyclic AMP is being converted into 5' AMP. Again, keep one thing in mind, whatever is being produced in the body, it has to be degraded once its function is over. If anything is retained in the body, it will lead to a disease. So when ATP is forming cyclic AMP, the name of the enzyme is adenylate cyclase. And when cyclic AMP is being converted into 5' AMP, the name of the enzyme is phosphodiesterase, or it is also known as PDE. Now see what is happening is whenever there is decreased concentration of glucose, or that means the body is undergoing hypoglycemia, this cyclic AMP sends the message, and this cyclic AMP simply makes protein kinase active. It makes protein kinase active, which was earlier inactive. Because it is activating something, no? So it will make this protein kinase active. Now, I told you that body requires glucose now. So obviously just imagine which process will be accelerated. It has to be glycogenolysis. And if we talk about glycogenolysis, the active or the regulatory enzyme is glycogen phosphorylase. So what happens is this glycogen phosphorylase, which was inactive, will be made inactive. So if glycogen phosphorylase is active, chapter is over. Glycogen will be broken down to form glucose. So that means the concentration of glucose will increase. Okay. So you can just simply see that, it's all like, it's like a cascade of reactions. Whenever there is decreased concentration of glucose, it will give instructions to cyclic AMP to convert protein kinase into active form. When protein kinase is made active, it will convert glycogen phosphorylase into active form, thereby breaking this glycogen to release glucose. And this is under the influence of GPCR, G protein coupled receptor. This we are going to again discuss in detail. This you are going to study in physiology, what are the different type of receptors. So this is G protein coupled receptor. right? And we also know that whenever the concentration of glucose decreases, which is the hormone which is released? Yes, it is glucagon and epinephrine. So glucagon and epinephrine are going to accelerate cyclic AMP. Okay? And if glucagon is accelerating, insulin has to inhibit. So insulin inhibits this. So I can simply say that insulin will inhibit glycogenolysis and glucagon or epinephrine are going to promote. Up the question is, the level of glucose comes back to normal. If this continuously keep on going, the body will undergo hypoglycemia. Okay, one thing. Up now here if I talk about if glycogen phosphorylase is made active, as you can see here, glycogen synthase now needs to undergo inactivation. So I can say that if glycogen phosphorylase is active, so glycogen synthase, I am just writing it as GS, so glycogen synthase will now, which was active earlier, which will be now converted into glycogen synthase inactive. Like this. Like this. Okay, 1 second, this will be inactive. Because only one thing can remain active na. Agar glycogen phosphorylase is active, glycogen synthase will be inactive. Now if we come over to another concept, concentration glucose ka it is increasing. Now this cyclic AMP needs to be degraded into 5' AMP. Now everything will reverse. That means this cyclic AMP will now be converted into 5' AMP. So if cyclic AMP is being converted into 5' AMP, the concentration of cyclic AMP needs to be decreased. So I can simply say that this will be decreased, right? This is a very important point to understand. Initially it was high, now the concentration of cyclic AMP will decrease. Now if the concentration of cyclic AMP decreases, protein kinase will be made inactive. Glycogen phosphorylase will be made inactive. That means glycogen will not be broken down. This will decrease. And this glycogen synthase will be now made active. So I'll just highlight it once again. Like this, and like this. Or overall, if glycogen synthase is active, we can simply say that glycogen will be produced. So the concentration of glucose will come back to normal, and that is why we say that this phosphodiesterase is activated by insulin, whereas inhibited by glucagon or epinephrine. So this is the entire cascade of reactions where we can simply correlate the two things, ki how this covalent modification is directly related to the hormones. So if I have to just visualize the entire thing, I can simply say that insulin is promoting glycogen synthase, right, which is active in the dephosphorylated state. Dephosphorylated state. On the other hand, glucagon is accelerating glycogen phosphorylase, where glycogen phosphorylase is active in its phosphorylated state. Very, very important. Similarly, epinephrine is again going to accelerate glycogen phosphorylase, which is active in its phosphorylated state. This is a very important concept which you have to understand. Now the question is, what is the difference between the mode of action of glucagon and epinephrine? So always remember glucagon only acts in the liver, whereas epinephrine acts in the liver, but the most important role it plays in the muscle also. This is the basic difference between epinephrine and glucagon. And if we talk about again the structures, I can simply say that this is actually serine. Glycogen synthase and glycogen phosphorylase, if you look at the structures, this is like serine is present at the active site. So it is occurring in two stages. OH is there and the second is OP is there. Right. OH can be converted into OP with the help of kinases. And OP can be converted into OH by phosphatase. So it can be kinases also I can write and phosphatases. And this becomes the structure of both the enzymes, whether it is glycogen phosphorylase, whether it is glycogen synthase. So if I say this is dephosphorylated, as you can see it is without phosphate. So dephosphorylated. And if we talk about this, this is phosphorylated. Now, if we talk about dephosphorylated and if we have to compare the structures, what would be the difference in the structures? Dephosphorylated means this is the active form of glycogen synthase and inactive form of glycogen phosphorylase. On the other hand, phosphorylated mein active form will be of glycogen phosphorylase and inactive form will be of glycogen synthase. So I can simply say that for dephosphorylated to phosphorylated, phosphorylated to this, it is none other than covalent modification. And that is why I told you these two are interrelated also, because covalent modification is conversion of OH to OP and vice versa, whereas insulin is going to act here. Insulin will make glycogen synthase active in its dephosphorylated form, whereas glucagon and epinephrine will make this into its active phosphorylated form. And one thing always keep in mind, insulin being an anabolic hormone, whatever processes insulin accelerates, the rate limiting enzymes of that particular thing are always active in their dephosphorylated form, be it PDH, be it glycogen synthase. Okay. So I can just summarize everything if I have to talk about the level of hormones. Insulin always promotes hormones. Yeah, so insulin is a hormone which always promotes the rate limiting enzymes which are active in their dephosphorylated states. Related form. An example is the most important example we just quoted now, glycogen synthase. And other enzymes we have to quote, I can simply say pyruvate dehydrogenase, because these are the two enzymes which are like pyruvate dehydrogenase. Very important. Glycogen synthase and PDH. And on the contrary, if we talk about glucagon, glucagon is going to activate which are those hormones which are active in their phosphorylated state. So I hope now you have understood the difference between phosphorylated and dephosphorylated form. An example is simple. The most important example is glycogen phosphorylase. So if somebody asks you a question that Glycogen phosphorylase की active form is what? So you have to immediately respond that active form is in phosphorylated form. Active form of glycogen synthase is in dephosphorylated form. And same is with epinephrine because they go hand in hand. But the only difference is glucagon acts in the liver, whereas epinephrine acts in the liver as well as the muscle. So this is important. Active in phosphorylated form, example is glycogen phosphorylase. Other examples are also will be there, which we will be discussing. Now this is covalent modification or hormonal regulation. Second is role of calcium. Now if we talk about role of calcium, always keep one thing in mind. In exercise, or you can say anything related to the muscle, calcium will always be released and it will always bind with calmodulin type of protein, and what's its function is it will make glycogen phosphorylase active. So if it is making glycogen phosphorylase active, that means it will give energy. And that is what exactly happens. Calcium always accelerates glycogenolysis, thereby leading to the increase in the concentration of glucose. So this is how this glycogenolysis and glycogenesis is regulated. Now if we have to quickly revise the gist, if I talk about in the liver what is happening and in the muscle what is happening. So what is happening actually is this is glycogen and this is glucose. Glucose can be synthesized to form glycogen and glycogen can be degraded to form glucose. So here glycogenesis is there and here glycogenolysis is there. Now if we talk about the differences, I can simply say that it is triggered by glucose 6-phosphate, whereas here it is inhibited by glucose 6-phosphate. It is inhibited by glucose. It is inhibited by ATP. Right. So obviously we know that whenever we have to synthesize glucose 6-phosphate will accelerate it. But whenever the concentration of glucose 6-phosphate increases, it will inhibit. Moreover, if we talk about glucose again, glucose itself will be inhibited because the body requires glycogen now. So there is no question of breakdown of this. So this is occurring in the liver. Okay. And if we talk about the muscle, what is happening in the muscle? The most important difference in the muscle and this is glycogen, glucose, glycogen to glucose, glucose to glycogen, everything is same. The only difference is glycogenolysis. Glycogenolysis is always promoted by calcium, promoted by AMP, right? Obviously inhibited by glucose 6-phosphate. Here we are not going to take glucose because glucose is not produced in the muscle. So no glucose as the muscle lacks glucose 6-phosphatase. On the other hand, here we can simply say glucose 6-phosphate will accelerate. So this is the basic difference between the regulation between the liver as well as the muscle. Okay. And if we have to again summarize this, I can simply say that here glycogenesis is there and here glycogenolysis is there. Now you should be able to remember all the basic terminologies in this, that glycogenesis, anabolic process, glycogenolysis, catabolic process, occurs in the well-fed state, occurs during fasting, or the instantaneous mode. So I would say glycogenolysis in the liver, then comes gluconeogenesis. Third important point is the rate limiting enzyme of glycogenesis is glycogen synthase. And the rate limiting enzyme of glycogenolysis is glycogen phosphorylase. Then insulin promotes glycogen synthase, whereas glucagon promotes it. So glucagon will inhibit it, whereas here insulin will inhibit it. And if we talk about calcium, calcium will always promote glycogenolysis. So this is, or you can say that, and one more point, yes, glycogen synthase is active in its dephosphorylated form. This is a very important concept which you have to understand. And here glycogen phosphorylase is active in its phosphorylated form. So if you have understood this concept, this thing will be very simple for you to understand. So here we finish up with the glycogen metabolism, and the most important point, which again I am focusing on this, is that glucose 6-phosphatase is absent in the muscle in a normal individual, because after this video we will be discussing about the GSDs, glycogen storage disorders, where you will see that there is a very important disorder in which glucose 6-phosphatase is present in the liver also. But overall, in a normal individual, glucose 6-phosphatase is absent in the muscle, and that is why Cori cycle has a very important role to play in the muscle. So this is all for glycogenesis and glycogenolysis, or overall glycogen metabolism. Thank you so much.